So in this video, I would like to talk a little bit about bacterial staining. It's an important topic as far as microbiology is concerned. So I've decided I will discuss this topic divided into two parts. And this will be part one of the two-part series. Here, I will touch on the basics of staining, and also I will talk in detail about gram staining. So without further ado, let's look at firstly main types of staining. So if we are talking about staining in microbiology, obviously gram stain would be number one, because most of the bacteria there are take up gram stain. So we can detect bacteria and make our diagnoses using only gram stain in most of the cases. But a few bacteria, fungi, or protozoans that do not take up the gram stain, well, for them we have acid-fast stain or some other stains that I will talk about in the next video. But for this one, let's just stick to gram stain. If we look at this picture, well, it's an interesting picture because we don't see only gram positive or gram negative. Actually, we see both gram positive purple colored cocci and also gram negative red colored bacilli. So it's a mixed culture slide. It's not a pure culture. So before we can talk about gram staining or any staining for that matter, let's just look at the basic structure of bacterial cell wall. We don't need to understand all of the structures here. Rather, let's just concentrate on the peptidoglycan layer. On the left, we have a gram-positive cell wall, and here we have a gram-negative cell wall. So the main difference that is noticeable between the two bacteria is the thickness of the gram-positive peptidoglycan layer and the negative peptidoglycan layer. So the gram-positive bacteria has a significantly thicker peptidoglycan layer. So, and this information will factor in when we talk about the differences in staining. For example, the gram-positives will retain the primary stain, that is the crystal violet stain, whereas the gram-negative will take up the counter stain and they will stain red in the end. So next up, a few terms that we should know before we can talk about the actual steps in the gram stain. Well, firstly, primary stain. And to make it easy for us to remember the why we use primary stain, I have added this piece of word here. So P for primary and P for pierce. So the goal of primary stain, remember that the primary stain has to pierce through not only the gram-positive cell wall, but also it has to penetrate through the gram-negative peptidoglycan layer. So as a result, we will get both the same colored cell wall when we apply the primary stain. There will be no differentiating factor when we are using primary stain. And when we use a trapping agent, well, the name suggests what it does. So the trapping agent or mordant actually makes the stain stick to the peptidoglycan layer more strongly. So we will talk a little bit more about trapping agent as we move on. Now let's look at a few examples of primary stain. So in case of gram stain, we will use crystal violet as the primary stain and carbofuchsin for the acid-fast staining. And as a trapping agent in gram stain, we will use Lugol's iodine solution. So actually, what we want to get here is the iodide ion from the iodine solution. And we'll talk about it a little bit more as we move on. So the few more terms of decolorizer. Well, like I said, the primary stain is used only to stain both the bacteria or both of the types of bacteria the same way. So we use the decolorizer to differentiate between the two types of bacterial cell wall. As the name suggests, again, it's pretty self-explanatory that decolorizer will try to decolorize the cell wall. And it's pretty intuitive to think that the thicker cell wall won't want to give up its primary stain. So the decolorizer will almost be ineffective in case of the thicker gram-positive cell wall. So the final step, counterstain. Well, I've written that counterstain is used to confirm what type of bacteria it is. So actually, after we decolorize, then we have to do a counterstain to recognize the gram-negative bacteria to be specific. So again, a few examples here. Ethanol or acetone are used as decolorizer in gram staining, and acid alcohol is used in acid-fast staining. And SAFRANIN would be our counterstain of choice for gram staining, and methylene blue will be used in acid-fast, which we will talk about in the next video. Now the actual steps, right? So in gram staining, the goal is to differentiate between two types of bacteria, whether the bacteria is gram-positive or it's gram-negative. So as I said, first we will use a primary stain to pierce through both of the cell walls, and in this case we will use crystal violet, and both of the cell walls will dye blue or purple. And then to make the stain more permanently stick to the cell wall, we will use a mordant. And again, in this step, there will be no differentiation. Both the cell walls will still remain blue. What happens actually is the iodide ion forms a bond with the crystal violet. So the complex, the iodide crystal violet complex, is more strongly embedded within the layers of peptidoglycan. And which one has more layers? Well, obviously gram-positive has more layers of peptidoglycan. So which of the cell walls will actually retain the primary stain more strongly? Well, obviously the gram-positive will. So moving on with decolorizer, now we try to differentiate between the two, as we know that until now both of the cell walls are purple. So when we use the decolorizer, acetone or ethanol, it's an organic solvent, and what it does is it tries to take the iodide and crystal violet complex out of the cell wall and tries to decolorize the bacteria. So in case of gram-positive, the decolorizer won't be useful, and in case of gram-negative, it will be able to take the stain out of this thin peptidoglycan layer, and the gram-negative will then look colorless. So now, how do we visualize the gram-negative that has become colorless after this step? Well, that's why we will use a counterstain. So blue and red, they are complementary opposite colors. So that's why we have chosen the SAFRANIN as a counterstain. So we can differentiate, like in this slide, the gram-positives are bright, vibrant purple, and the gram-negatives are bright, luminous pink. So it's a very clear distinction, right? So that's why to visualize the gram-negative cell wall, we use a counterstain, SAFRANIN, and the end result is we get blue or purple gram-positive bacteria and pink gram-negative bacteria. So now to summarize the gram stain, well, before that, let's look at a few examples. Well, here we see these are round-shaped bacteria. So these are cocci, and on top of that, these are forming chains, right? Short chains. These must be Streptococcus, especially that because we see that the bacteria here are dyed purple, and we know the gram-positives dye purple. So they must be gram-positive chain-forming cocci, and well, what's that? Well, that's Streptococcus pneumoniae. And on the right, we see here these are rod-shaped bacilli, and they are red-colored, so they must take up the counterstain, so they must be counterstain SAFRANIN, so they must be gram-negative. So in this case, they are gram-negative bacilli, and very famously, these are E. coli. Now to summarize, let's look at this chart. It's something that will help us remember the information more easily, right? In a more organized way. So primary stain, crystal violet, and both of the bacteria stain purple. Then we use a trapping agent to trap the primary stain within the cell wall. And again, both the bacteria will still be purple. And then we try to differentiate, right? D for differentiation. So we try to differentiate between the bacteria, and we use alcohol or acetone as the decolorizer, and the gram-negative, having a thinner peptidoglycan layer, will be decolorized, so they will be colorless at this step. And then to visualize them, we will use a counter stain, a red dye named SAFRANIN, and then the gram-negatives will dye pink, whereas the gram-positives will still be able to retain the primary dye, which is the crystal violet. So the end result would be blue or purple colored gram-positives and red colored gram-negative. So that's mostly it for this video. Now, to give you the absolute high yields, well, obviously you have to remember these few steps and what it actually does. Most of the time, what happens is we recall that what primary stain we use and what trapping agent we use, and we memorize these stuff, but we tend to forget why it is that we use this stuff. So that's why I have created this simple mnemonic type of device for you guys to remember it easily. And also, the most important thing would be to retain in the long term. So remember, the primary stain pierces through both the gram-positive and negative bacterial cell wall equally. Then the trapping agent traps the primary stain inside the cell wall, and the decolorizer tries to differentiate between the gram-positive and gram-negative cell wall by trying to take the color out of the cell wall, and it's successful in case of gram-negative because it has a thinner peptidoglycan layer. And then to visualize that gram-negative bacteria, we use a counter stain to confirm that whether the bacteria is a gram-positive or a gram-negative. If the bacteria takes the counter stain, then it will be a red gram-negative. And if it doesn't take the counter stain, well, obviously it will then be blue colored gram-positive. So here I'd like to conclude my video, and in the next part I will continue on with talking about the acid-fast or Zeal-Nielsen stain, and also a few other important high-yield stuff. So until then, well, keep learning and keep sharp.